afternoon, everyone. I'm François Boissel. I'm the CEO and co-founder of Novadiscovery, a professional services company specialized in silico clinical trials. We support biotech and pharma partners in optimizing the research and development of new drugs. Now we do this with a unique methodology and an engine which enables effectively the upstream prediction of a drug candidate's clinical benefit, potentially years before the first administration in humans. This works as follows. We combine a mathematical and computational model of a disease of interest with a drug candidate, as well as a virtual population representative of a specific jurisdiction, for instance, the French melanoma population. And we de facto offer our partners a digital sandbox where they can test multiple assumptions cheaper and faster than would otherwise be the case with more conventional in vitro and in vivo assays. They can better inform their decision making and certainly optimize the design of their clinical trials, notably by reducing the number of patients to be enrolled, by ensuring that we specifically identify the optimal responders. So effectively, this approach generates benefits for our clients through an increased probability of success, or conversely, the early detection of failures in the making before too much capital is being deployed. We also reduce costs on absolute terms, as I've mentioned, for instance, in reducing the number of patients required to run a trial, and we also speed time to market by exploring a number of assumptions much faster than would be the case with more traditional approaches. This approach is standardized in a sense that it is applicable to any sort of therapeutic area, and it is really applicable from early discovery down to the completion of clinical development. So the whole idea is that we build over time an in silico asset for our partners, which will enrich as the program we support progresses, and which will really serve, again, as a digital sandbox to test any number of assumptions in a much more reliable way than the more conventional assays. So we go through the target selection, obviously looking at the understanding and elucidating innovative drugs' mechanism of action thanks to these comprehensive mathematical models of disease processes. We also point to potentially the right animal models, target the right indication, optimize the drug candidate's regimen, and identify and characterize in as specific as possible a way the responder profiles to increase the chances of a successful completion of clinical development. So it's all about reducing costs, improving probability of success, de-risking decision making, and it's also about this learn and confirm paradigm, which is emerging in the regulatory agency's mindset, and I'll go through a couple of points on this particular issue in my next slide. The agencies, and the FDA and the EMA in particular, are gradually seeing in silico clinical trials as something that will eventually become used on a systematic basis ahead of all the more conventional assays. So it's not a matter of substituting in vitro and in vivo assays within silico, of course. It really is a matter and a function of improving decision making by testing a lot of assumptions in a more reliable way, informing those upcoming assays, and then feeding back data and information gathered through those assays into the model so that it gets refined and more reliable in terms of predictive capabilities, while the ultimate validation obviously is the confronting the in silico phase 3 trial with the actual in vivo trial outcomes. Now, in terms of opportunity, we currently operate a professional services model, but as the market and the industry matures within the next few years, we anticipate serving actually two separate markets. A professional services a business model targeted towards biotechs, So this would typically mix an element of retainer and success fee, and potentially forming joint ventures to support early stage assets in order for us to capture as much of the value we create as we can. And the separate market will become eventually big pharma players, and we would address those through a software licensing of our technology platform, a business model. Now, in terms of the regulatory agency's mindset, again, I've stated that they are making now commitments to the in silico approach, and this was evidenced by a couple of things on the FDA front over the past few months. First of all, the first guideline on model validation for dossier submission purposes was released by the FDA in last September, a year ago. Over the summer, the US Senate and the House voted a bill giving an explicit mandate to the agency to move forward on these approaches, and essentially stating that these in silico clinical trials have the potential to save up to $100 million on any given research and development program. Those agencies are basically engaged in, again, maturing the regulatory science around in silico clinical trials. Now, the magic formula in order to predict this drug candidate clinical benefits upstream is actually fairly straightforward to understand. This is what we have called the effect model of a drug candidate. This was discovered and developed by our co-founder and chief scientific officer, who was actually instrumental back a few decades back in the emergence of the randomized placebo-controlled phase 3 trial, which is nowadays the gold standard. It works as follows. You basically apply a disease model to a virtual population of patients. All of these dots that you see here are individual virtual patients. For all of these patients, applying the disease model will give you on the horizontal axis the probability of suffering from a particular clinical event of interest. So here, for instance, this is a use case on testing a drug for the prevention of lung graft rejection without the treatment. So that's effectively the control group. Then the second step consists in adding to the disease model the drug candidate submodel. Apply this back to the same virtual patients, and this time around you get on the vertical axis the probability of the same clinical event of interest, but this time around modified by the drug candidate, which effectively means that for each virtual patient, we are in a position to quantify the expected clinical benefit from the drug candidate, which is simply the arithmetic difference between those two probabilities. Now, we call this the absolute benefit, and the further away the patient sits from the line bisector, the larger the expected clinical benefit. And if you sum all of those absolute benefits over the entire virtual population, you get what we call the number of prevented events, which serves as a population-wide efficacy metric we then use to run our optimization scenarios. So if you were to approach Nova asking, for instance, what is the optimal dose for my product, we would answer by testing thousands of different doses, but identifying the most promising avenues by looking at those scenarios maximizing the number of prevented events. And this is, again, a very standard framework that actually works irrespective of the therapeutic area. Now, this use case was a project funded by the European Commission a couple of years ago, where thanks to the ability to specifically characterize those optimal responder profiles, we were able to optimize the design of a phase 3 trial, bring it back from 3,000 patients using conventional sizing approaches to 6,000 patients, which effectively could translate in significant savings. Another interesting feature about this effect model, as you would have guessed by now, is that each patient is her own control, so you're effectively removing bias with this approach. Now, this is a very summary illustration of the workflow as to how we build our disease models. The primary material we use is actually knowledge extracted from the scientific literature. So effectively what we try to do is to represent all the biological mechanisms involved in the disease of interest. If we look at modeling, for instance, colorectal cancer, we would have cell proliferation as one of the submodels, apoptosis as another submodel. So for each of these subcomponents of the model, we would review the literature in order to identify all the biological entities involved, describe their functional relationships, and this is really important to understand. We're in a causal paradigm. Map them and represent them first in a textual format using the first module of our platform, of our engine, which is called GitHealth, which is a collaborative environment dedicated to extracting knowledge from those scientific articles. We would then go through a validation process, which I'm happy to discuss with you after the presentation. We would then convert this into mathematical formalism. So each of these knowledge models, for instance, again, the apoptosis, would be converted into mathematical equations, then converted into computer code. Now we're entering the second module of our WISE engine, which is SimWork, which is programmed using Haskell as a functional language, a very scalable language. And once we get the disease model, the drug submodel, and the virtual population, we're in a position to run the in silico experiments to answer the questions that were asked. Now, a key difference versus some of the much hyped machine learning approaches we hear a lot about throughout the industries these days, our belief is that using data as a base material for predictive purposes is not reliable for a number of reasons. The first reason is that human biology is eminently complex, and those data are extremely time and context dependent. So each and every time you'll use a particular data set to try and derive a predictive algorithm, chances are when you deploy the algorithm in a different jurisdiction at a different time point, the algorithm won't be that predictive. Second of all, those algorithms are based on correlation analysis. Even the most sophisticated machine learning approaches, at the end of the day, this is correlation you're looking at. This doesn't tell you much about the actual causal relationships, whereas our approach, which may be more resources intensive to deploy, actually maps all the functional relationships between the biological entities. Last but not least, they operate as black boxes. Our approach is fully traceable. You can go from a simulation output back to each primary source of information that was used thanks to our WISE engine. It's fully traceable, fully auditable. Sure, I'm almost done, perfect. So in terms of the therapeutic areas we've been applying this approach to, we've been working on cardiovascular diseases, we've been working in cancer, lung fibrosis, hepatitis B and sepsis, covering a number of applications from animal to man transition support, and in particular elucidating the reason why 300 neuroprotective agents failed in clinic after promising results on rodent models in acute stroke. The explanation was unfortunately fairly simple, but without a model, you wouldn't have encapsulated everything that was known at the time in terms of the biology of the disease. We've been working on optimizing phase 2 and the phase 3 trials. So again, the key message is it's very flexible and adaptable. And we're looking for a 10 million Series A. We expect to close the funding round around Q2 2018. And this will serve to expand our sales team and especially establish a footprint in the US market, accelerate the development of our technology platform, those two modules that I briefly mentioned, GitHealth and SimWork, scale our library of cancer models and virtual populations, as well as strengthen our senior management. Thanks.